about organic molecules. Okay, so you'll need that packet out. And I think let me just kind of remind us of what we've done so far. So we've said that we've got carbohydrates. We said we've got lipids. We've got nucleic acids. And I'm just reviewing here, so don't need to write this down. And we've got proteins. Okay, and so we know that we have 4 functions for carbohydrates. We have energy, and we have the structural, and we have sugar phosphate backbone of nucleic acids, and then we have cell communication, and we've listed several things for these. We've listed glucose and glycogen and other monosaccharides and disaccharides as 4 possible energy things. We've listed cellulose for structural. We've listed RNA and DNA, ribose and deoxyribose for sugar phosphate backbone, and we've listed glycoproteins for the cell communication. And we've actually done a little bit of understanding how structure reflects function. Remember, we've talked about on glycoproteins, we have these chains, these oligosaccharides, oligo meaning many, so anywhere from 3 to about 20, these chains that hook into each other and connect with each other from different cells and allow cells to identify each other. So this is a means of cell communication. Then we talked about lipids. We started talking about lipids, and we talked about the first category of lipids, which are fats, and these are energy supply. So for energy, we have fats, and the fats are made up of glycerol and 3 fatty acids. Okay, and we've also talked a little bit about how fats can be saturated or unsaturated, and what the significance of that is medically, heart healthy versus unheart healthy, and what the significance of unsaturated fats is as far as for animals like fish, for example, that live in cold waters. Their cell membranes are made up largely of unsaturated fats because those fats don't congeal and get solid at colder temperatures, whereas animal fats do. And we said that animal fats that are saturated have a risk for heart disease, and unsaturated fats like vegetable oils and fish oils are not going to be heart unhealthy. They're going to be heart healthy. And so that's where we kind of left it. And I think we were about to start talking about cell membrane, right? Okay, so why won't you shut?
We're on page 20.
Had we gotten to 26?
We're on. Yeah. We haven't filled in the columns.
Okay, so let me just remind us here. Cell membranes, phospholipid bilayer. Our second function then related to lipids is that we're going to be talking about cell membranes. And we remind ourselves that because this is all hydrocarbon, it's very, very, very nonpolar, and it doesn't mix well with water. So there's this one part of the molecule that actually is a little bit more polar, but for the most part, this is almost entirely nonpolar, and it doesn't mix well with water. That's important to us because we want it to be our cell membrane, right? So remember, we also said that water is very polar. The way that it mixes with and dissolves things is by positive regions attracting negative regions of the solute molecules, and its negative regions attracting positive regions. And so if I have a water molecule, and I'm just going to draw this here real quickly. If I have a water molecule, remember that it has more of a positive end and more of a negative end. The more negative ends near the oxygen, the more positive ends near the hydrogen. And if I'm trying to interact with other things out here, other particles, and I'm trying to dissolve them, it depends on these other particles also having positive and negative charges here. So if I have a negative charge on this particle right here, I can develop a force of attraction. And the water molecule literally acts like a tractor beam, and it pulls those pieces apart. But if I don't have any charges, if I'm really nonpolar, water doesn't have anything to latch onto and pull into solution. So it can't mix with water. And so the fact that water's polar and hydrocarbons are nonpolar means they can't mix or dissolve with one another. So would it make sense then that if we wanted to create a barrier, come on. Okay, to keep the fluid outside of a cell, in other words, extracellular fluid, and the fluid inside of the cell separate, okay, would it make sense that we would construct it of a material that doesn't mix well with water? That would make sense, right? Okay, so maybe even those fat molecules that we've been talking to, with a slight modification of the fat molecule, we can accomplish this goal. And I want you to follow this pattern of thought here. If we simply use the fat molecules to try to form cell membranes, because fats and water don't mix and fats are less dense in water, what's going to happen to fat molecules if we try to use those to make them a cell membrane? If I mix the fat molecules with water, what's it look like the fat molecules are doing?
They're going to move together.
They're just going to float, right? They're floating on the surface. So they're just floating on the surface. Okay? That doesn't help us much if they're just floating on the surface. Okay? That doesn't help us a whole lot. So what can we do about that? Let's structure reflects function our way into something else here. Maybe there's a way that we can modify the fat molecule so part of it becomes polar and mixes with water, and the other part stays nonpolar and forms the barrier. And so to do this, we're going to modify the fat molecule into something that we're going to call a phospholipid. Okay, don't get nervous. I'm not going to ask you to know a chemical structure here. Okay, so the first thing we're going to do is get rid of one of the fatty acids. Okay, we're going to get rid of one of the fatty acids and take that off. And then we're going to add on something that contains phosphorus here. And this phosphorus group also contains a lot of oxygen and it contains nitrogen. And because I've got lots of oxygen and nitrogen here in this phosphorus group, that makes that end of the molecule very polar. So this end of the molecule is going to mix with water. This end of the molecule is not going to mix with water. Okay? So, if I scroll down then, we're just going to call that a P-containing group. So as opposed to the fat molecule where we've got 3 fatty acids, we're going to have the phospholipid, and that's where this name comes from, this phosphorus-containing group here, and the rest of it being lipid. And we can just visualize it like this. We've got this phospholipid here. But you can still ask, well, how does that help us? And I would say, again, the presence of the phosphorus group along with lots of oxygen and nitrogen make that end very polar, and that end mixes really well with water. So the fatty acid end is nonpolar and doesn't mix well, and this is often visualized as having a polar water-loving head and two nonpolar water-hating tails. So we're going to draw this structure right here. This is my phospholipid, okay? This is a polar water-loving head, and these are nonpolar water-hating tails. Does anybody remember what the term for water-loving and water-hating are from Sperry Young?
Hydrophilic is water-loving and hydrophobic is water.
Okay, so hydro meaning water, philic meaning loving, and hydrophobic meaning water hating. Okay? So those are terms that we'll need to be familiar with. Hydrophilic and hydrophobic. Oops, my parentheses kind of came. So I've also got that description here, and you can see how these relate to each other. But you might ask, and this would be a legitimate point, won't these molecules also just float on the surface of water, with the water-hating part kind of sticking up in the air and the water-loving part kind of mixed in with the water? Okay? That's certainly a likelihood if there are only a few of these molecules. But if I get a bunch of these molecules in water, they do something that I think is really pretty special and amazing. If I dump a bunch of these phospholipids in water, if enough of them are close enough to each other, they actually form a two-layered spherical structure. Okay? And they do this by themselves. It's a spontaneous thing. They don't require anything to put this in place. They create this bubble. Okay? So on one side, I've got the water-loving heads on the outside facing the water, mixing with the water, and the tails towards the center. And on the other side, I've also got the water-loving heads pointing toward the water in the center of this, and their tails pointing towards the center of the layer here. And since I've got two layers of phospholipids, I call this a phospholipid bilayer. Okay, there's two layers, so it's the phospholipid bilayer. And that's spontaneous. Now in the cell, when I've got cells making other cells, there are actually things that cause this phospholipid bilayer to be created. It's not like we just dump a bunch of phospholipids in fluid inside of our body and it magically forms these cells. But these things do form spontaneously without the help of anything if you just dump a bunch of phospholipids. And it's really amazing, and you can see why this would be a good thing to be a cell membrane. So the arrangement of phospholipid hides the parts of the molecule that hate the water, but they expose the parts that love the water. Okay, so we're always going to show our phospholipid bilayer like this. That's kind of our model, except what I would probably do if I'm just drawing this is I'm going to go something like this. Just kind of, and another one on the other side, and I'm just going to just kind of scatter in these fatty acid tails as well. So I'm not going to draw this out in great detail like this. I'm going to make it look a little bit more like this if I'm just drawing it really quickly. Okay, so we've got the polar heads and the nonpolar fatty acid tails. Okay, so we'll talk a lot more about membrane structure soon, but for now, I just need you to remember the basic structure of the phospholipid and how that basic structure is different from the structure of a fat. So this is our cartoon of a fat, glycerol plus 3 fatty acids. This is our cartoon of a phospholipid, glycerol, 2 fatty acids, and a P-containing group. And I'll ask you to be able to draw those for me on a test. Okay. The third type of lipid we said we wanted to talk about are steroids. Steroids are a class of chemical messenger called hormones, which are substances that are released under certain circumstances to travel to different parts of the body and then cause one or more specific actions to occurring depending on what the message is. Okay, so I can have one hormone, and again, I'm not going to yell at you and scare you like I usually do and cause you to release adrenaline, but adrenaline's a pretty amazing substance. Think about it. The way that it causes so many different things to happen is that it attaches to so many different cells. Adrenaline goes to your heart muscle and causes the heart muscle to speed up. It goes to your nervous system and it causes the respiratory system to speed up. It goes to your temperature center and causes you to increase your temperature. So it does all kinds of things to get you ready to fight or flee. And it's just one hormone. But what makes it do so many different things is the number of different cells that it actually goes to. Okay? So it causes one or more specific actions to occur depending on what that message is. Okay? All steroid hormones are based on the structure of the cholesterol molecule. And notice the letters S-T-E-R. Cholesterol. And this is where we get the term steroid. Steroid is a hormone. A steroid hormone is one that's based on cholesterol. Okay, so I've shown you the structure of the cholesterol molecule. You don't need to know this. It would be nice if you could recognize this, but this is the structure of the cholesterol molecule, and it's all carbon and hydrogen. This is, again, a very non-polar molecule. It's got 4 hydrocarbon rings. Again, you don't need to know that structure, but it's almost entirely made of carbon and hydrogen, and it's very nonpolar, and it doesn't mix with water. Okay, so what types of chemical hormone names are you familiar with that have that STER term in it, or that you think might have the STER term in it? What have you heard of? For example, what's the male hormone? Testosterone. Testosterone. There's the STER. Okay? How about there's a female hormone called progesterone? Good. Progesterone has the STER in it. Okay? Even estrogen doesn't have the STER in it as STER, but it has ESTR, doesn't it? So all of these terms imply that we've got the steroid hormone or the cholesterol molecule as part of that hormone. So all we do is we just change a little bit of each of these molecules and it becomes a different hormone. Okay? The only thing I need you to remember about steroids at this point are they're one of 3 types of lipids that we talked about. Instead of being composed of a chain of carbons, they're formed of rings of carbons, and they're made from cholesterol, and they are chemical messengers. You don't need to know anything more about the structure. Okay? Now, Hopefully there's some white space still left on on that back page. Go to that back page. Do you still have some space left?
No.
No? Okay, then I'll need you to get out a sheet of paper because I need you to write some stuff down.
A raw piece of paper.
You're welcome. You're welcome.
I have, well, that's not.
I thought you were going to do. Good.
I have a couple of pieces of paper up here if anybody. I have 3 pieces of paper. Anyone else need a sheet of paper? Okay, so, no, I don't want to say that. So, next time when you come to class, you're going to write down the next version of this: carbohydrates, lipids, and then nucleic acids, and proteins. We haven't added any nucleic acids or proteins yet. But again, you're going to write down for me that we've got energy, we've got structure, we've got sugar phosphate backbone, of nucleic acids, and if you want, I will let you abbreviate this SPB of NAs. That's probably better. Huh? And then we have cell communication. Okay, so there's 2, 4, 6, 8 points with the main names. There's 4 functions there, so that's 4 more points. And now we're going to add the lipid functions. I'm going to say energy. That makes sense. And we're going to add cell membrane. That makes sense. And we're going to add steroids. Okay? And we remember that I've also offered you some extra credit if you can remember the individual names of these molecules: glucose, and then glycogen. Oh no. I'm running out of disk space. Crap. Glycogen, and then other mono and disaccharides.
It's monodisaccharides?
I put a slash there, mono and disaccharides. Yeah, mono and disaccharides. Okay, so that's 4 quarter points, and then structure is cellulose, and then sugar phosphate backbone, ribose, RNA, deoxyribose, DNA, and then cell communication was glycoproteins. Hope you can read my writing there. And then for energy for lipids, we're going to say fats. That's all we need for that. For cell membrane, we're going to say phospholipid bilayer. Bilayer. Remember I showed you that bilayer, right? Phospholipid bilayer. And then for steroids, we're just going to say cholesterol. Okay. So that's your question of the day for next time. So that's 2, 4, 6, 8, 9, 10, 11, 12, 13, 14, 15 points, and probably 3 additional points of extra credit. Again, you'll see this on a test. So if you don't do well on these question of the days, the test can make up for that and give you extra points on your quizzes if you do well on the test. Okay. Now, I know this seems like a lot, and it's a lot of memorization. It's not necessarily a lot of memorization if you have a strategy of about how to think about this and simply just learn the ways in which these things function. And if you have a good visualization of what some of these molecules look like, you can see that they function in a certain way. For example, the phospholipid bilayer, if I look at that, it just makes sense that it's a cell membrane, right? It looks like a cell membrane. The fats look like storage molecules, okay? So if we try to connect these things with images, that'll help us remember these different functions. And it's important for our understanding of the physiology. So any questions about this? Is there anything up here that is illegible to the point that you need me to spell it out again? Yeah.
So to reiterate, steroids are hormones that are built upon cholesterol.
Correct. Okay. And like I said, we just changed one little part of it, and that one little change means that it can't bind to certain cells and cause certain actions to happen, or it can bind to certain cells and cause actions to happen. We'll talk a little bit more about how molecules cause cells to do certain things. That's part of our physiology. Okay? Any other questions about the list here? Okay. So we're going to need to stop this for just a minute. I didn't realize this was going to be a complication. Talk amongst yourselves. Okay, so I'm back up here. Okay, so anyway, hopefully you remember from biology that the way that this works is that the code gets read in the nucleus, so that gene there represented by wherever that red dot is what gets read, and then I send a piece of data out to the ribosomes to create the factory. What's that piece of data called? Does anybody remember? It's a message, and that should help. We create what kind of molecule from the gene? Messenger RNA? Never heard that term before?
mRNA, yeah.
mRNA, yeah. You've heard of mRNA? So messenger RNA. So that's what this little snaky thing is here. This little purple thing is the code that goes to the ribosome, and then the ribosome reads that code and makes the protein based on that. Okay? This is knowledge that you have to have related to how a cell works. Okay? So the nucleus contains the DNA. The DNA is the code, and the code gets read when the cell needs that protein, and then the nucleus makes messenger RNA, and it's called messenger RNA because it's carrying a message to the ribosome, and then the ribosome reads that message and makes the protein based on that. Okay, so the RNA is messenger RNA. The ribosomes are also made of RNA, so that's where the RNA part of it comes in. Okay, so the DNA part of it is the code. So the DNA stores the code, and the RNA transmits the code. Okay, the RNA takes the code to where the code can be used. Okay, so that's the protein code part of it. That purple color is kind of pretty. The other thing is that the DNA transmits hereditary information, right? I take the DNA from one cell and copy it and put it into another cell. So that's the other function of the DNA, or the nucleic acids. The first function is to be the code. The second function is to transmit hereditary or genetic information. Okay? We need to know something about the structure of these nucleic acids. We need to be able to understand that in order to understand genetics. Okay, so nucleotide consists of 3 parts. A 5-carbon sugar. That's weird. Let's not use that anymore. 5-carbon sugar.
Let's select the next one.
So a 5-carbon sugar, okay, a phosphate group, and a nitrogen containing group. Okay, we have a 5-carbon sugar, a phosphate, and a nitrogen containing group. So what about the 5-carbon sugar? We're going to look at each of these pieces. We're already aware of one carbohydrate or sugar that's glucose. That's a 6-carbon sugar, and it looks like a hexagon, right? This is what I will expect you to be able to draw for me related to glucose. Just a hexagon with an O in it, and then a little stem off the top. Okay? That's my cartoon for that. Okay? The sugar molecule that forms the central portion of a nucleotide has 5 carbons. And it's a molecule shaped like a pentagon. Okay? Just like with glucose, we number the carbons. We start with the oxygen and we go 1, 2, 3, 4, 5. Okay, so all you'll have to do to be able to draw this is that. Okay, just like the glucose, that will be my expectation for you. Notice the carbons are numbered. Notice there's an incomplete bond at the first carbon, and this is where the nucleotide base is going to attach. The nitrogen-containing base is going to attach there. And then notice there's an OH group attached to the third carbon here. Okay? And this will be used as a handle or a connector to attach to something else. Also notice that there is an OH group attached to the 5 carbon. Okay, so I'm going to tell you how to think about these things. As with glucose, we'll not always need to present this 5 carbon sugar as this complex of a molecule. I may use 2 other forms of this. And this is the form that I'll expect you to be aware of for this sugar. So we need to have a name for this 5 carbon sugar. But let's remember the general class of the names here. We haven't stated this yet, but you're probably aware there are 2 main types of nucleic acid. We have stated that in the past. We have deoxyribonucleic acid or ribonucleic acid. And so you're aware that the Na stands for nucleic acid, right? Okay? So you don't have to memorize that. You already know this name, nucleic acid. Are we getting close to time? No, we've still got more. So you don't have to remember that. You just know that Na stands for nucleic acid. So if you remember the name of this macromolecule, you already know 2/3 of the name, and you just have to remember what the D and the R stand for. So the 5 carbon sugar is called ribose. Right? Ribose for ribonucleic acid. And then DNA is deoxyribose. For the full name of deoxyribonucleic acid. Okay, so there's nothing magic about these names. The only thing you have to remember is the name of the sugar, ribose, deoxyribose, and then nucleic acid. Okay, one other thing to notice here then is what's the difference between the two. On the left-hand side, there is an OH group at the 2 carbon, and this is ribose. On the right-hand side, there is no oxygen here. So that should make sense. Deoxy, we've taken the oxygen away. We've deoxygenized it, right? There's deoxyribose and ribose, okay? So ribose and deoxyribose, and that's the difference. And those will actually cause some changes in the way these molecules can form different structures. Okay? So once again, deoxyribonucleic acid, remember nucleic acid because that's the name of the type of molecule. And we remember deoxyribo because deoxyribose is the sugar. Ribonucleic acid, we remember ribo because ribose is the name of the 5 carbon sugar. Now what about the phosphate group? This is a molecule that simply is a phosphorus surrounded by 4 oxygen atoms. I don't need you to know there's a double bond there, but I have a phosphorus surrounded by 4 oxygen atoms. We won't go into great detail about its chemistry other than to say that it is another way that we link things together. Okay? Oxygens are going to link things together. So in the nucleotide, the phosphate group is typically shown as a circle with a P in it attached to the 5 carbon. Okay? So, how difficult is it going to be to remember that this 5 carbon sugar is a pentagon that looks like this? That's not too terribly difficult, is it? Then I know that with that carbon there, I'm going to connect to the next one through the phosphate, and through the carbon, 3 carbon, I'm going to connect to the one below it. Okay, so that's how I'm going to connect my sugars together, and that's why this is called a sugar phosphate backbone. Let me draw it a little smaller here. Okay, sugar phosphate, sugar phosphate. Okay, and it's just one long chain. Sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. 3 billion times. The DNA molecules in your cells are about 3 billion of these in length. Okay? So if you can imagine 3 billion of these, sugar phosphate, sugar phosphate, sugar phosphate, 3 billion times, you can imagine the DNA code. Okay? Well, and it's like, well, Marshall, that's not very much like a code. It's all the same thing. Sugar phosphate, sugar phosphate, sugar phosphate on infinitely. But then remember, we had this other thing that sticks off the side here, and this is where the letters of my code get attached. And we're just going to pretend that that's a box. Okay, we're just going to pretend that that's a box. And I have 4 different ones of these. Those are the letters of my code. I have 4 different boxes that I can attach there. Okay, so here I've just shown you again how these things attach to each other. Sugar, phosphate, sugar, phosphate. And that's not really that difficult. It's all based on this number 5 here. And the action is at the 5 and the 3, where they're connected to the phosphates, and then the rest of the action is at the 1 carbon. So all of the action is at the odd numbered carbons. The nucleotide base is the foundation of the nucleotide. It's part of the nucleotide that tells us which one of the 4 possible code letters it is. It's fairly complicated. We'll learn a little bit more about it when we talk about protein synthesis. I'm not going to show you the actual chemical structures at this point. All we're going to do is call it an N-containing base right now. And let's put all of these pieces together and how I want you to see this and be able to create it for me. We're just going to start with a pentagon with an oxygen and a stem, and then we're going to put our 5 carbon there, and then we're going to put our phosphate group here, and then we're going to put our N-containing base here. Okay, that's our N-containing base. And that's a nucleotide. Okay, does anybody remember what the 4 different bases are? The names of the 4 different bases?
What does the box say?
N-containing base.
Okay.
N-containing base. Does anybody, yeah? What are my 4 letters of my code?
A, T, G, and C.
Okay, and A stands for adenine. G stands for guanine. C stands for cytosine. And T stands for thymine. Not thiamine, which is the vitamin, but thymine, which is the nucleotide base. These are the 4 letters of my code, right? And all DNA really is 3 billion of these things lined up, and I might have an A, C, A, T, G, T, C, A. This might be a string of DNA, right? And how many of these, do you remember how many of these code for a particular amino acid? When I sit, yeah, 3 at a time, right? Okay, so this would code for a particular amino acid. This would code for a particular amino acid. This would code for a particular amino acid. So again, when I'm in the nucleus here, I create this message that's this DNA right here, this RNA, and it's got these codons, and we'll talk more about that when we talk about inheritance, but we have these 3 bases here. Each one of these codes for a different amino acid. So when this gets to the ribosome, it gets fed into the ribosome, and the ribosome reads, and it says, okay, now I need to put this amino acid in. Okay, now I put this amino acid. Okay, now I put this amino acid in. And that's how we build our proteins. We create a chain of amino acids based on that. Okay, so it sounds like it's at least familiar to one of you, and probably you as well for these.
Are amino acids always a trigroup of the 3 of the 4?
No, this is just the code that says put the amino acid, the correct amino acid into the chain. The amino acid, remember, is a structure that looks like this. We talked about this. An amino acid has this structure. Right? Remember, the COOH group is the acid part, and the NH2 group is the amino part. And then we have 20 different ones where this part right here is different for each amino acid. So I have 20 different R. It's like a variable in algebra, right? And it could mean 20 different things. But these amino acids, so this group right here might code for this amino acid. It might say, okay, ribosome, grab that amino acid and put this into the protein. And then this one says, okay, grab this other amino acid and put this into the protein. And this one says, grab this third amino acid and put this into the protein. Except it happens at light speed. It's unbelievable. We'll watch some videos about how fast this actually occurs. We can make proteins with hundreds of amino acids in them in minutes or seconds. Okay, so let's stop at that point. We'll finish this next time. We'll talk about proteins. Next time you just have the question of the day with the carbohydrates and the lipids. We'll add the nucleic acids and proteins into it next time. And then we'll be done with the chemistry.
That's good. Best part.